we're still waiting for one committee member, but in the interest of time, I think we'll get started. And, so, welcome to this very special day. We're very proud to have Ipshita finish today with her PhD, and I'll tell you a couple of things about her that you may not know, and I'm trying to not embarrass you. So. But so many may not know how accomplished Ipshita already was before she joined UCSD. She won the National Talent Search examination, and she was then at the Berlin Institute of Technology and Science in India, and she was one of the top 1% students out there. And she held a fellowship that it's hard for me to pronounce. So. And during that time, while she was at Berlin, she won three. She was selected for 3 international exchange programs, one in the US, in Germany, and one in Switzerland. And I think that's where I met her for the first time. I don't know if you remember, but I had the pleasure to do the Skype interview for the graduate program. So, and I'm glad I recommended her for being accepted in the program. And we were all very glad that she decided to join. So she joined the PhD program in 2013. And she joined the lab in 2014, and as I was just mentioned a moment, one of her first projects was her work on grid cells. And just a couple months after Ipshita joined, we were all very excited to hear, that the Nobel Prize was awarded for the discovery of grid cells and plate cells. So it was a big event for our lab, and we found out beginning of October of that year. But only a few days later, we found out that Ipshita's uncle won the Nobel Peace Prize. So we felt, at least indirectly, very accomplished. So Ipshita's first project, I think she'll talk to you more about the science, but I'll tell you a little bit about that background. She took over a project in which we stimulated long-range projections from the megaseptum to the campus. And that project had been pretty anemic for a couple of years before she joined. And we were very impressed to see that she got the whole thing on track within just a couple of months. And it took some time to write it all up. But one of the details that you will not see when she presents these data is that during the revision, we essentially redid the whole study. So we actually recorded more data in that study during revision than and we is Ipshita in this case. So she recorded more data during just 3 or 4 months than in the entire a couple of years before that. So, and then Ipshita's core project, as you will hear, was on mechanisms that result in the generation of grid cells. And I think we all think that grid cells are so fascinating because they really tell us, that the brain generates cognitive concepts. And in this case, we can actually very easily visualize these concepts by recording cells via distinct patterns. And I think what's particularly groundbreaking about Ipshita's work on grid cells is that it's really the first that has identified the computations that lead to the generation of grid cells in the medial spinal cortex. So, and she received the HHMI International Student Fellowship for proposing that project. and she really transformed the lab in many respects. We were before that a traditional electrophysiology lab. Now we are capable of doing a whole range of techniques that I can say pretty much all Ipshita introduced into the lab. And so you will hear, that Persa is quite innovative, so innovative that we had troubles publishing because I think the reviewers really did not appreciate that you are several years ahead of your time. And finally, as everyone who knows Ipshita knows that she's an extremely humble and unassuming person, and she is a great communicator. She is really effective in mentoring people, and what's probably not measurable is her impact she made on all the people around her, and so that's I think something that I'm particularly proud of. And last but not least, you all know that Ipshita won a couple of awards for PhD work already, and so she won the Graduate Student Research Excellence Award and also the Founding Faculty Award. And for the Founding Faculty Award, we have the honor of having a picture hang right next to the Nobel laureates of the division. So we will have at least some memory of your time, in addition to all the ones that we had the privilege of making. And with that, I would welcome the family in India. It's a very special occasion, and please, Ipshita, looking forward to your talk.
Thank you so much, Stefan. I've been watching lots of thesis defenses online when I knew I have to present, but also people in the past, and I always imagined I would start crying. I don't know. I kept thinking of all these emotions I would have, and right now I just feel numb. So I guess that's good in a way. But thank you so much for the introduction, and there'll be a good acknowledgement section in the end. Stick around. So good morning. Today I'll be talking about my project. I'm not going to read out my title. It's way too complex. I should have something simpler. But what I will start with is the tale of the mouse who loved burpees. And so some of you may have heard the story once before, but I'm going to repeat it. So burpees are this Indian sweet that you can see over here. And usually we eat these burpees during Diwali when we celebrate. And so over Diwali we had all these guests over at our apartment. And I went to open this box of burpees and I see this little mouse sitting there chewing. And I live in Mesa Graduate Housing, just as a disclaimer. So I see this mouse there, and then we have all these guests in the house, and then I was like, okay, we need to do something about this. So it turns out I'm terrified of mice that aren't in a plexiglass cage. I didn't know that. So I asked Sachin here to set up a trap with the burpees and catch the mouse, and so we managed to catch the mouse. And so this is where I live, over here. And we figured we could just drop off the mouse somewhere over here, and that should be fine, end of the day. So we dropped it off. The next day, I opened the box of burpees, and I see the mouse sitting there again. And I knew it's the same mouse, because the mouse had one eye. I'm sure it's the same guy. So again, he was smart enough to find his way back home, but he wasn't smart enough to learn the trap. So we caught him again, and then I didn't know what to do. So I put him in a bag, took him on the UCSD shuttle, then released him next to Bonner Hall, and gave him access to the loss. The question that we're addressing over here is how did the mouse actually know how to get back to the bird feeds? And it turns out that the brain actually has a very well-developed navigation system within this region that you can see in this fancy animation called the medial temporal lobe. And now the medial temporal lobe has several other brain regions that I will gloss over today, but the two most relevant for this talk today are the entorhinal cortex and the hippocampus. And the reason we, and as Stefan mentioned, the studies on these two brain regions were awarded the Nobel Prize. And the reason this is so exciting is that if you look at neural activity within these brain regions, the neurons there are actually tuned and display really interesting features that I'll display right now. And so in the hippocampus, you have these cells that we call place cells. And in a second, you'll understand why we call them place cells. So over here, you see a rat running around an open field. In gray is its path. And the red dots are every time a single neuron in the hippocampus turns on or fires. And so you can see the animal is running around for a while. And what you can hopefully appreciate towards the end is that this neuron chooses to fire only in this location. And because it seems to prefer this certain location, these types of neurons are called place cells because they fire in a certain place. And we can visualize this as a heat map, which is some of the images you might see later in the talk, where in red are regions where the neuron fires more, and in blue are regions where the neuron does not fire. Okay, so in the hippocampus, we have this whole network of cells called place cells. Why am I interested in the entorhinal cortex? Well, if you look at the activity of cells in the entorhinal cortex, there are certain cells that we call grid cells that fire in, to me, even more interesting manner. And so this is a similar video when an animal is running around an open field. Every time a single neuron again fires, you can see white dots. And so as the animal is running, there are white dots. Every time the neuron turns on. In the beginning, it doesn't look like there's much happening. But if you wait for a while, you'll see that this neuron actually fires in a very regular pattern that forms a hexagonal grid, which is why these neurons are called grid cells. And I wait for the video to finish. So these are so beautiful and exquisite to me that I think, which is why I think a lot of people are attracted towards understanding how grid cells function. And so now you can imagine that in this medial temporal lobe, in the entorhinal cortex and the hippocampus, we have a network of cells that are firing as grid cells and place cells and other spatial cells that I actually haven't mentioned yet. And so if we think about this scene back to my house and where we dropped off the mouse, probably the mouse used his network of grid cells and spatial cells in the entorhinal cortex. Shown here are just two cells that are tied in the entire environment, forming an hexagonal pattern, and his network of place cells in the hippocampus to actually find his way back to my house. And so in this talk, I'm going to divide my talk into two parts. In the first part, I'm going to talk about this network in the entorhinal cortex and talk a little bit about grid cells. And then next, I'm going to talk about place cells and how they might be firing and how their firing is controlled in the hippocampus. And so this is the first part of my talk. The title of my summary is that recurrent circuits within medial entorhinal cortex support grid cell firing. And so as I just mentioned, I've been talking about grid cells this whole time, but there are also a lot of other spatially tuned cells coexisting with grid cells in the medial entorhinal cortex. For example, we have head direction cells, and these are neurons that only turn on when I'm facing a certain direction. So for example, a single neuron will turn on only when I face this direction, but not when I turn around. And so you have thousands of these head direction cells also within the entorhinal cortex. And you have speed cells that increase their firing rate with increasing running speeds. And together, all of these different cell types are thought to provide a metric for navigation. Now, the medial entorhinal cortex is a 4 to 6 layer structure. And what's interesting is that while head direction cells and speed cells are found across all the layers, and in fact, they're actually found outside of the entorhinal cortex too. The reason I find grid cells the most interesting is because they're highly enriched within this layer 2 of the medial entorhinal cortex. And in fact, you can rarely find them outside of the MEC, and MEC will be the medial entorhinal cortex, so you can rarely find them outside, but most of the time you only find them within layer 2. And so let's think a little bit about these grid cells. So here's a grid cell, and imagine an animal starts with two different trajectories, one along this way and one along this way. Now, I hope you appreciate that the neuron actually needs to receive a lot of information in order to fire correctly for this to happen. So its firing must be tightly controlled with constant updating with respect to the external environment. So the neuron somehow needs to receive the information that the animal traveled along this trajectory and is now here, versus traveling along this trajectory and is now here. And because these neurons are most enriched in layer 2, the question that my project is trying to address is, does this hexagonal firing arise from connections within layer 2 of the entorhinal cortex? Right. And so to do so, we turn towards transgenic mouse techniques in order to be able to target specific cell types within layer 2 of the MEC. And to do this, we used a WFS Cree mouse line. You can see in this beautiful image in a cross, where we cross the Cree mouse line to an AI14 reporter line. And this mouse line labels pyramidal cells that exist as patches all along the dorsoventral axis of the medial entorhinal cortex. And again, as you can see in the schematic, it specifically labels only layer 2, but not other layers of the MEC. And so if we do some immunos to confirm the identity of these neurons, when we can double label with WFS1, you can see that there's a really good overlap with Cree expression and WFS1 expression. And so using this, we can layer 2 pyramidal cells are exclusively targeted. Okay, so we know a little bit about where these cells are, but let's find out a bit more about where they project. And so to do so, we use the help of Dr. Brilliant, who's here, and the brilliant viruses that Arad and he have been making. And so this virus allows you to express the red fluorophore NRUB2 in cell bodies and axons, and GFP, which is tagged to synaptophysin, at the axon terminals. And so when we perform an injection of this virus into the MEC, you can see that the cell bodies are labeled in red, and there are dense green axons all along layer 2, but not outside layer 2. And if we look outside of the MEC, you can see axons within C1, but rarely except, apart from these projections, there are hardly any other projections to other regions of the brain. So we thought this is a good mouse line to be able to target layer 2 of the entorhinal cortex and actually perturb the activity to then test how different functional cell types behave in response to this perturbation. And so we performed optogenetic activation. By this, we express channel rhodopsin unilaterally in the medial entorhinal cortex, where by shining blue light, we can then activate the cells every time the light shines on. And at the same time, we could record from the medial entorhinal cortex while performing this perturbation. And the animals were just run on an open field for 3 sessions with a stimulation performed in the middle, flanked by baseline sessions. And so now I'm going to show you the activity of some of the neurons and how they responded. And for an example, I'll show you this peristimulus time histogram. This is a single neuron. And you can see when the red is when the light turned on. And you can see in response to the light, this neuron shows a really nice activation that's quite consistent over trials. And so this image, this single image can be summarized as a single row in this heat map, where all neurons that showed any kind of excitation are arranged as single rows. And the darker colors, the warmer colors imply that the neuron was activated or had a higher firing rate. And so these are cells that were only activated. If we look at the remaining cell type, not the remaining cell types, but if you look at the remaining responses, you can see there are several cells that show this peak of excitation, but also this really long dip of inhibition. You can also see this in the deep blue over here. Similarly, you can see cells that don't seem to show excitation, but only get inhibited, visualized over here. And then some of the cells, very few of them, seem to show no or a very weak response. And so when we summarize this across all the mice and all the layers, we actually find that what's known by the anatomy, what I just showed you, and also through slice electrophysiology using past experiments, is that these pyramidal cells, the ones that I'm targeted, project within layer 2, but hardly project to layer 3. But what they do is project to interneurons within layer 2, which subsequently probably inhibit all other cells around them. And so what we find is consistent with this knowledge that within layer 2, you see a good amount of excitation followed by an inhibition, whereas in layer 3, you mostly only see cells becoming inhibited. So as I just mentioned, these cells, these pyramidal cells, are actually excitatory in nature. And so we wanted to look a bit more to confirm why we see this inhibition. And so what we can do is actually classify our cells based on their waveform and their firing rates as interneurons, which are the inhibitory neurons, or principal cells, which are these excitatory cells generally. And you can see, according to the color code, that most of these interneurons labeled over here do show excitation shortly after every light pulse. And consistent with this idea, you can see that even in terms of the latencies, if you look at the latency for excitation, it comes first, followed by the latency for the inhibition. So basically what we're doing is we're turning on this pyramidal cell network, everything turns on, then the inhibition kicks in, and everything shuts off for a period of time. And this inhibition is really important to us and what we'll be using for most of the project. So again, we have this network, we have a channel rhodopsin during photoinhibition, population of cells is inhibited. And the question that we're trying to address is, once these neurons recover from the inhibition, how do head direction, speed, and grid cells respond to this perturbation? And to give you a simple analogy of what this might mean and what I'm actually trying to do, let's imagine a system of Christmas lights. And so over here, assume that these lights are actually sending a very important message that is purely coincidental and has nothing to do with today. So if we do assume that there's suddenly a power surge and the light goes off and there's a power cut, and suddenly there's a pause in the system. The sequence is stopped, the cells aren't firing anymore, sequence is in progress. There are several possibilities of how this might continue. One, the lights can just start and resume right from where they paused. Two, the whole sequence could just start again right from the beginning. And three, it could just start turning on random letters. And each of these different possibilities implies a very different role for this network of lights, which is an analogy for our network of neurons and how they might be performing the sequence that is being displayed. And so again, what we're asking over here is, how does this inhibition affect the firing properties of these different cell types? And so I'm going to start with speed cells over here and talk a bit about what we found about them. So if we actually look across the entire session, the baseline 1, stimulation, and then baseline 2, over here are 3 examples of speed cells, all responding very differently to the light pulse. And you can see that overall their speed tuning doesn't seem to be really changed by a lot. It's more or less constant and it doesn't seem to be disrupted. So then next we looked at how these cells are actually responding to the light. And so shown here are similar heat maps from what I showed you before, but now ordered according to what their responses are. And you can see again that a lot of the speed cells do show this profound inhibition or excitation, so they are being perturbed by our manipulation. It's not that they're not being affected at all. And so next we can then address how these speed cells are affected right at the period of inhibition, similar to the analogy I showed of turning off the lights from a set of Christmas lights. And so we can look at spikes that happen right over here in the green section, which is when the cell is being inhibited. And we can, as a control, take an equal number of new spikes in this baseline pre-session. And we can compare the speed scores of spikes happening specifically here versus here. And what we find is that there's a reduction in speed tuning under both conditions. It's not beyond what would be expected in the controls. And so let's think about this for a second. I just told you that the speed cells are actually cells that increase their firing rate with increasing running speed. So if we do a manipulation where we're actually inhibiting a neuron, it's obvious that if we decrease the number of spikes, the speed cell will not be able to effectively have any kind of speed tuning anymore. And so what we're doing over here, we think, is that inhibiting the neuron by itself, reducing the spikes, is decreasing the speed tuning, but it's not any further than what would have been expected by chance. So what we believe is happening over here is that this network, at least, is not further impairing the speed tuning of neurons. Okay, so next we can look at how head direction cells respond to this perturbation. Oddly enough, we found again that head direction cells don't show any impairment in their head direction tuning. But what's more interesting to us is we found that you don't even see the really precise inhibition that we were seeing with all the other cell types. And if you remember the proportion of cells that I showed you do not get inhibited, that's a really small proportion. And most of them were these head direction cells. And so these head direction cells seem to be a part of a separate network coexisting with all the other cells, but not receiving much of the local feedback connections. Next, we wanted to examine if even during these short time periods when the rest of the network is inhibited, what's happening to head direction cells. And if we examine it, then we can see that there's no impairment in the head direction tuning, consistent with what we would have predicted. So head direction cells seem to be fine. What happens to grid cells? Over here, you can see 4 examples of grid cells, all again responding very differently to the perturbation. And overall, you can see that the grid tuning actually seems really precise over the entire session. There are cells like this that get really strongly activated, and that just increases the background noise, but the overall grid tuning and the firing remains quite preserved even with the perturbation. Next, if we examine at how the grid cells are responding, again, you see this large, long inhibition that stays in the grid cells. So these grid cells are receiving a lot of the local feedback inhibition that kicks in with this manipulation. So now we can examine what happens again at these shorter time periods, the times when the cell is actually inhibited, and how the neuron recovers at this time point. And so shown over here are the spikes that are occurring during the green section, plotted over here, versus the spikes that are occurring in the pink section, which is a control, the same number of spikes plotted over here. And what maybe you can appreciate, it took me really long to actually see this. For 2 years, I thought that there was no impairment in grid cells, and that was the story we were going with. But what you can see is that there actually tend to be more spikes occurring outside of the grid field during the inhibition versus the control. And you can quantify this and see that there is indeed a slight increase in out-of-field firing during this window of inhibition. So why is this effect so small? Why can we barely see it? And we think it's because that if we actually take windows further and further away from the inhibition, this error rapidly goes down. And so we think there's this error correction that happens within 10, 15 milliseconds that corrects any kind of mistakes that happen in grid cell firing on a moment-to-moment basis. And so we find again that grid cells do seem to show a bit of impairment, but rapidly regain their properties. And this is important because grid cells do need to rapidly receive constant information from external sources. So probably this does make sense. And so together, to summarize this part of my talk, we find that in this network, within the entorhinal cortex, we have head direction cells, grid cells, and speed cells. And the grid cells and the speed cells specifically do receive feedback inhibition. But when it comes to the firing properties, consistent with what we would have predicted, because head direction cells and speed cells do exist outside of the entorhinal cortex, we believe that their tuning properties are merely inherited from outside of the entorhinal cortex. Whereas in case of the grid cells, we believe it's a combination where there are effects of the local circuit and the local computations. But in addition, there's also constant resetting from external sources. Okay. With that, I'd like to now talk about part 2 of my talk. So over here, I'll talk a bit about the hippocampus and face cell activity. And so this is the first project actually that Stefan was mentioning that I worked on. And I have to thank Mark Brandon. He was the postdoc in the lab when I first joined, and he's the one who actually taught me everything. In fact, when I joined the lab, I was terrified of the lab because I didn't understand a single thing we were doing. And he helped me move from that to what I am now. So I'm really grateful. Okay, so as I mentioned, all this time I've been talking about spatial tuning. I've been talking about grid cells, about place cells, head direction cells, speed cells, but honestly, sometimes spatial tuning is not enough. And so to give you a better idea of what I mean, I'm going to show you a small schematic. So imagine there's this animal that's running along the track. And as he runs, there are 5 place cells in the hippocampus that turn on, right? Now there's cell 1, cell 2, cell 3, and all of these cells have overlapping fields. So as it moves along, this one starts, and then this one starts, and so on. Now, obviously, such firing does give you some amount of resolution of where you are in location. So if we compare these 2 zones, you can actually see that there are distinct cell populations that signify these zones, and so you can actually resolve a certain amount of location. But there's a certain resolution to this, and so if you go beyond your scale of resolution, you've completely lost any information about where exactly in space you are, because you have the same set of cells firing once again. And so we believe that neural oscillations can provide a solution to problems such as these. This is just one example, and of course they do a lot more. So what are these neural oscillations that I'm talking about? With reference to the hippocampus, these oscillations are called theta oscillations, and they're thought to arise from this brain region called the medial septal area. And so if you record from electrodes in the hippocampus, or actually in the entorhinal cortex, you see this large prominent oscillation in the 6 to 9 hertz range, recorded in the local field potential. And lesions of this medial septal area drastically impaired this oscillation, which is why people believe that the septal area is responsible for a large part of these oscillations. And so why am I talking about these oscillations? Well, the firing of hippocampal place cells is tightly coordinated by these theta oscillations. And so shown as the schematic I showed you before, again, this animal is running along this track. It has 5 place cells, and you have these ongoing theta oscillations. And what the key factor over here is that most hippocampal place cells oscillate at a frequency that's slightly faster than this ongoing local field potential. And so I'm going to show you in a second what that means. So let's assume the animal enters the field over here. This oscillation is going on, and the red cell fires, right, over here. Now, because it's firing at a frequency slightly faster than the ongoing LFP, the next spike will happen at a slightly earlier phase of the oscillation. And what I should say is that every time the animal fires, the first few spikes tend to occur in late phases of theta, so towards the end of the oscillation. And so as it progresses, the spike happens earlier and earlier, and so on. And so this is for the red cell. Similarly, you can see it for the yellow cell, the blue cell, the pink cell, and the green cell. Now, hopefully what you can appreciate is that because of this happening within a single cycle, the order in which the place cells occur is retained. So even on a time scale of a few milliseconds, you always have a sequence of progression that's telling you precisely where in space you are. So, okay, so this is important. We have place cells that are strongly tuned to theta oscillations. And we have these oscillations, the local field potential around at 8 hertz, and we've got these cellular oscillations that tend to be a little bit faster, around at 9 hertz. And so the question of this project is actually to try to address how these ongoing oscillations influence the cellular oscillation frequency. Are they coupled? If we change one, does the other one change? Can we try to examine this a little bit more? And so the aims of this project was first to try to alter the frequency of theta oscillations, next to address how phase field properties are affected by when we do this perturbation. And finally, the most important is how is the oscillation frequency of the hippocampus cells changing when we change this local field potential oscillation. And so what I did mention before is that the medial septum is thought to generate these oscillations, but mostly the frequency of this oscillation is thought to be controlled by the GABAergic cell population within the medial septum. And so in our manipulation, we used PD-3 mice to actually target these cells, the GABAergic cells within the septum, express channel rhodopsin within the septum, and put an optic fiber over the medial septum in order to turn these cells on and off. At the same time, we recorded from cells and local field potential in the hippocampus. And the behavior was really simple. The animal was just running around a circular track. Every time the animal crosses the zone, the light turns either on or off. So light is delivered at either 10 Hertz or 12 Hertz. We chose these frequencies because they're both slightly outside of the endogenous theta range. And so they give us a good control to look at how the cells are responding to this perturbation. And just alternate laps, light on, light off. And so I'm going to show you what the local field potential looks like when we do this perturbation. And so this is the same video, but slowed down much more. Over here, you can see the raw local field potential trace. And this is the filtered within the theta range. And you can see that right now, there are these ongoing theta oscillations at 8 Hertz. And the instant the animal crossed the zone, the blue light turned on, and the oscillations immediately shifted to the stimulation frequency, which was in this case 12 Hertz. And so the animal runs along the length of the track, reaches the end of the track. This frequency still persists. The 12 Hertz light is going on, it's chugging along. The LFP is chugging along. The animal reaches this end. It's about to cross the zone again. And you can see when it crosses the zone, the endogenous theta oscillation returns to a slower baseline frequency. And so with this manipulation, we can do a really precise and rapid control of the frequency of oscillations to whatever frequency actually that we want, either 10 Hertz or 12 Hertz. And so over here you can see in this session, the animal ran several laps on and off. This is a 10 Hertz session. Over here is a heat map showing the power in the local field potential. So over here during baseline, you have most of the power within 8 to 9 Hertz, and during stimulation here, you have most of the power at 10 Hertz. It's more easy to visualize here, but during stimulation, you have most of the power at 12 Hertz. On and off, and this is repeated over several trials. Okay, so we can alter the frequency of theta oscillations, which gives us a really powerful tool to now test how these oscillations influence cellular properties in the hippocampus. So next we can address how phase cells are measured by altering this frequency. And for the interest of time, I'm not going to show you the data today, but what we found is that though the spatial properties do remain intact, we actually found that the blue light turning on and off was causing the cells, the phase fields to remap. And for those who don't work with the hippocampus, to remap just means that when you move an animal from one environment to a different environment, the whole population of phase fields changes to represent the new environment. And so the blue light was actually causing the animal to perceive it having been in a new environment. And so do you have control is the story. But then finally, we can now address how the oscillation frequency of the hippocampal cells is affected by doing the pacing. And what I'm showing you here is an autocorrelation of a cell. Basically, it just shows you the rhythmicity of a neuron and how it's firing. So this is one cell, this is the second cell, this is during baseline, and this is during 10 Hertz stimulation. Sorry, that should be a 12 Hertz stimulation. This is during 12 Hertz stimulation. And this just shows you the power in different frequency ranges at which the neuron is actually firing. And you can see that for this neuron at baseline, it prefers to fire around 9 Hertz. And then during stimulation, it shifts to a 10.9 Hertz preference during the 10 Hertz stimulation. For 12 Hertz, this neuron is firing around 7.7. During stimulation, it shifts to a 12.7 frequency. And so these are a few neurons. I can show you all the neurons over here. So again, this is a heat map with each row organized as a single neuron, and in red is its peak oscillation frequency. And in the black dots are the LFP frequency at that time, and so you can see there are a large number of neurons that are firing at a frequency slightly faster than the ongoing LFP frequency. And you can see during the 10 Hertz stimulation session, a large number of these cells shift their oscillation frequency beyond the stimulation frequency, in this case at 10. And at 12, a smaller proportion, but a few cells do shift their frequencies to 12 Hertz. And what's important over here is that they not only shift their frequency, but especially in case of the 10 Hertz, they also accelerate their frequency beyond the local peak potential. So consistent with what I talked to you about phase precession, where the cells are firing at a frequency slightly faster than the ongoing LFP. And so we interpret this as almost like these septal neurons acting as a pacemaker, playing music, and that there are further cortical and intrahippocampal computations that then set the frequency of the neurons to not only precisely match the ongoing LFP, but rather accelerate it beyond so that it can actually continue doing all the other sequence functions and then consolidation functions. And so today, in summary, I've talked to you about two distinct projects. One within the entorhinal cortex, talking about how local circuits control firing of neurons. One within the hippocampus, where I talk about how long-range oscillatory circuits might be promoting coordination between neurons. And I believe these are interesting questions to study because I really believe that a thorough understanding of how neural circuits meet functional firing properties is the first step towards understanding how the brain is capable of generating complex behavior. And so the hope is that if we can understand how these circuits are wired and how they work, then we can actually start understanding how the brain works to some extent. And with that, I'd like to thank you for your attention. Thank you all for being here. I have to thank so many people in my lab. There'll be a much longer acknowledgement section after a round of questions. But so far, these are all the people that have been very closely associated with everything I've done. And thank you.
So I have a question just about the last pacing experiment. The fact that you looked at two different frequencies of pacing, 10 and 12 Hertz. And I guess I have two questions. One is, what determines or what underlies the fact that 12 Hertz, for example, seems to change the frequency of firing of a much smaller fraction of cells than 10 Hertz? And the second question is, what determines whether or not a cell is actually regulated by the pacing versus those that don't? You know, produce global manipulation.
Yeah. So we've actually done a lot of follow-up. So this was just the first part of the study. Since then, we've done a lot of follow-up to actually answer exactly those questions. So first, we did a lot of manipulations of 8 Hertz because we believe that's a more endogenous range and that can compare with the 10 Hertz. And we find a really strong frequency effect, where at 8, almost all the cells can keep up and be engaged. At 10, it's a smaller population. At 12, it almost falls off. But even this, the strength of this engagement is slightly dependent on our control of the theta oscillations, and we think that's dependent on the time and bias expression. So in some animals, so this is quite variable from animal to animal. And in some animals where we have much better control of the oscillations, we have more number of cells that can shift. So I don't think it's a matter of whether the cells are possible or capable of shifting. I think if we push them enough, they are. But I think they inherently don't want to. So unless they're really forced with very, very strong optogenetic activation of all neurons in the septum, they chug along really happily at 8, less so at 10.
Right, and I guess maybe the thing that I'm talking about is in an individual experiment, in an individual recording, you have neighboring. The cells are all experiencing the same amount of expression, right? And yet my understanding is some will be paced a fraction and others will not.
Yes.
I guess I'm surprised that you have this incredible heterogeneity, given the fact that I would think all the GABAergic input that you're regulating is
Yeah. Yeah, we honestly don't interneurons get tightly locked regardless. Even in animals with really deep spacing, actually, the interneurons are perfect. So maybe it's the intracellular properties of the neuron. We're trying to actually also generate a model now to try to predict a competition model to with single neurons to see how different inputs can cause a drive or replace or not.
Well, first of all, good job. I have a question regarding the first one to the talk, the response to the simulation, like a really combined diversity of responses. Like, what do you think contributes to like the diversity of activity and gene expression that actually
By diversity of responses, you mean some neurons aligned or not? I always assumed it's connectivity because we're not manipulating all neurons, we're manipulating specific subtypes. And so we know already that these neurons preferentially target certain cells but not other cells, certain interneurons but not other interneurons. So when I say we're perturbing this network, it's a little misleading because we're perturbing any network that's associated with these layer 2 parameter cells specifically, not necessarily the whole entire network. So to me, I think the heterogeneity arises because of connectivity.
great presentation. Fascinated by the first part particularly, and my question is whether you have an insight at this point into the error correction mechanism and any potential way to disable it.
So I think there are so many interesting experiments we can do with that. We know grid cells persist in the dark with few cues. So one experiment we could do is actually repeat the same perturbation but do it in the dark with very few sensory information to see if maybe the correction doesn't happen then. At the moment I can't really say that it's happening because of external inputs because the grid cells come back at the same time that the network, all the other cells in the endocrine cortex also come back. So I'm not sure if the input is coming directly to the grid cells or whether the fact that the speed cells and the head direction cells and all these other cells are now firing at the correct firing rate is playing a role. But I do think the experiment with very little sensory information or something where there's a sensory mismatch between the firing of the grid cell and like on a treadmill, for example, where you can perturb the network and then get an incorrect sensory information, then see there's more of an error.
So I would assume that at certain frequency, the intrinsic sequence of events, because frequency consolidation should be disrupted. And if that's the case, the hypothesis that the sequence is important for navigation, you see any problem again when you find the location.
Yeah, so this is not work that I've done. There's a graduate student in our lab who's been working on these questions. So we do find that the sequences, at least at 12 hertz, for example, would be impaired. And consistent with that, Dell has actually found that if you do a memory task, then you see an impairment at 12 hertz or 10 hertz, for example, but not at 8 hertz. And that's actually what motivated us to start recording at 8 hertz to see how the neurons are moving. And so we do think that we have some evidence to suggest that maybe sequences might be preserved still at 8 hertz, but not at 12, and that could lead to changes in behavior.
You mentioned remapping occurred. Was this during or persisting after the perturbation?
It's really cool. Right. Actually, I'm going to show you the remapping that happens from the GFP because it's the same as the channel rhodopsin. These mice were injected with an AAV GFP, not a channel rhodopsin, and we just the neurons. So as I said, there are 10 labs. So this is lab 1, on, and then off, lab 2, on, off, and so on. So you can see that this field, this cell has a field over here during the on labs, but this field changes to a different location during the off lab. So it's within a single lab.
With the GFP control.
Yeah. And then, so for example, this is more striking. We have all of these fields within this cell with the on labs, and then on the off labs, it just remaps. And it's really consistent. In fact, I made this plot because I found it really pretty, where it's just a correlation matrix of the different labs, and you can see like the even labs are the off labs, and the, like for example, 2 versus 3 has a really low correlation because the fields just completely.
Do you think this would be true with no viral injection also? That there's something to do with blue light?
So yeah, we did a lot of things to control for it. We started turning down the power little by little, and then there was a point where the power was low enough for it to not. We did try with the blue light. I think it's still blue light. And actually, it's funny. The blue and the green light led to similar fields. I can't distinguish so much.
If there's no further question, we can go on to the part. Did you have more to
I do.
show? Yeah.
I tried to keep it quick. I think I got too excited. Okay, so with that, I'm going to start this phase of my acknowledgements. I'll try to keep it short. Yeah, I'll try to keep it short. So Stefan mentioned that, as I said, I interviewed with him. I hadn't read any of his papers. I did not know. I'd actually, I think I was planning to, I wanted to rotate with Jill. And then I interviewed with Stefan, and it was like, oh well, easy, let me just set up this rotation. And so I rotated with him, and then honestly, I didn't understand a single thing I was doing during my rotation. I don't think I couldn't even read it and understand what it was doing. And then, so I joined him, and I was actually a terrible person when I was having this meeting, when I was telling him that I wanted to join his lab. I think I said something like, I want to join your lab, but I don't want to do the work that you're doing. And I think I cannot believe that he still took me after that. He stopped for a second. He was like, sure, you can do whatever you want. But then it eventually worked out to be a really fruitful collaboration with him and Stefan, and I had the freedom to do anything I wanted. I've learned so much from him. He knows everything. There are times when I feel really proud of myself and I don't talk to him, and then I just realize how much I don't know. And that's an amazing thing to happen. Actually, I think every student should have that experience. And I also really appreciate him for loving science the way he does, because I think I love science, but I love science in a selfish way where I want the experiments to work out the way I want them to. And I really admire how there are many times when I told him a certain result for months, and then after a certain point I'd be like, I'm sorry, I was wrong. This is not how the result is, and I'd be terrified, and he'd just be like, okay, so let's move on. And that's perfect, and I really admire that, and I hope someday I can be like that too. So thank you for teaching me everything. I've been lucky enough to not just have Stefan, but 2 other mentors, 3 mentors. Byung-Kook, he was the one who showed me all these fancy tools, and I was so excited by the science that he was doing. And all throughout, anytime I needed advice, I would just walk into his door and sit down with his notebook and ask me to draw schematics and things. And I really appreciate the guidance he put in me. I want to thank Jill for all her support, all her, as I said, like she's been across the hall, across the corridor, and talk to her anytime. Her feedback during lab meetings is extremely useful. I think I've learned how to get better talks because of her. And so I'm really grateful for all her input. And of course, I didn't have better pictures of my so I just took them off the internet and saw them. I really thank you for all your feedback and advice and also during my next, I want to thank this lab. This is an extremely nice photograph, which I'm sure you'll appreciate. And really, all of these people have been so helpful to me, and I'll talk a bit about them shortly. But firstly, I want to thank the people who I've had the privilege of working with and learning from, and they taught me so much about what it means to be a teacher. I thought I was a decent teacher, and then I started mentoring people, and I realized how difficult that is. Giselle was there when I joined the lab, and I didn't know what I was doing, and then I had to mentor her, and she didn't know what I was doing. But it worked out well. I really am grateful for her help. Stanley joined us recently, and he's an amazing. There are some students with whom you don't have to worry at all, and he just has this time. Your time will come in. Come in, you're like, okay, what am I gonna do? And then he just goes and does it, and then he leaves. And that's perfect. It's like ideal. It's what you want. He's really the ideal undergraduate. And most of all, I have to thank Leland. They tell you that you shouldn't become close friends with the people you work with, and I probably won't do this after that, but this time I'm just so grateful that I did become close friends with Leland. She's probably my best friend over here at UCSD in San Diego, and we've been through so much karaoke, posters, graduation, and she's just been phenomenal. And not only she's a great scientist, she has amazing work ethic. She has great initiative. She wants to take up projects on her own, and she's taught me so much about what kind of mentor I want to be. So I'm really thankful for her. Next, I want to thank all my lab. They've been so supportive. Stefan's lab has been sort of a girls' power lab, and I really like that. I don't know. So this whole group of people, here they are toasting donuts. Sananda, Claire, it's been amazing working with you too. And the people I really want to call out over here are Sylvia and Marta. They've been office mates with me, they're postdocs, and I've learned so much about being a scientist, but also about they're both moms of two very young babies. And earlier I used to think that if I wanted to do science, I just couldn't have. I mean, there's just no balance. And they made it look so easy that now I think it's actually maybe possible. And I admire them so much for everything and gossip about it. Stuff I know that as I said, I've been lucky enough to work with several labs. This is the LIM lab over here. Parties with them. It's been great working with the LIM lab because I don't have to do any of the work or the cleanup duties or the actual when things go bad. I just exit. And then, but I am around when there are parties and when I want to use the equipment and learn from them. So I would advise all of you to find a deal like that. And most of all, I want to thank Parag and Eric. Parag has been so useful, so helpful with providing viruses. And Eric helps keep me jaded about science, which is a good thing, I guess. He's telling me how awful science is. But I really appreciate him being around. He makes me happy. Just funny things with the corridors. I want to thank all the people at UCSD. Kavya, we were together in high school. We didn't talk to each other much then. And then we ended up here and figured we have to talk to each other. We've become pretty good friends. We watch movies together. Nandu has been my other part of India over here at UCSD. And everyone else, there are too many people here, so I'm not going to name them. But we have celebrate birthdays every week, and it means a lot to have an active group of colleagues and classmates with whom I can share troubles and discuss issues. So I would not have made it here without mentors who have guided me for before I actually came here. And so this is a shout out to all my previous PIs for having guided me and taught me what it means to be a scientist. And so this was my chemistry teacher back in high school. He passed away from cancer a year after I graduated high school. And he was the one who gifted me his book, Factors in the Brain. And this is the reason I'm actually in neuroscience today. So I'm really grateful for him for this book. All of these other mentors I've worked with, especially Dr. Ronald, he was the first. He was my PI when I first came to the US for a summer internship, and he had written this to me in a book when I was reading. And honestly, there are still days when I open it and I'm down, and it makes me feel better. He really motivated me to take up science and take up graduate school. I need to thank my friends. These are all of my friends from high school. I'm still in touch with them. I meet them. Many of them are in the US, especially I want to thank Mukta. She's probably flying over from DC right now. She couldn't make it today, but she'll be there tonight later to celebrate with me. I want to thank all of these people from undergrad. A lot of them helped proofread my SOPs when I was applying to graduate school. They meet with me. They just listen to me rant about graduate school, and I'm so grateful to all of them. One common feature you might be seeing a lot over here is this person. This one. And so this is Sachin. I've known him since I was in undergrad. You can see I've known him back in 2009 and on now. And he used to be my biggest competition. So I did undergraduate school. So there was this class in which he got an A, and I maybe didn't, or he got more marks than me or something, and I think I started crying. So we've come a long way from there. But honestly, he motivates me to be a better scientist every day. There are times when I come home and I just start watching Mean Girls or some stupid movie, and then I look over at his computer and he's just reading the latest issue of Euron. I just roll my eyes and then turn off the movie. But really, I mean, there have been so many times when I've gone home crying, and his love for science is, as I mentioned, not selfish like mine. And I really admire that. He loves science for the way it is, irrespective of the result. He feeds me when I'm low. I don't know what else to say. Thank you. Lastly, at the end, I want to thank my family, my brother, sister-in-law, mom, and dad. Let me introduce you to them. This is Dr. Zutshi, Dr. Nadehi, Dr. Lata, Dr. Zutshi again. So hopefully you realize why I did my PhD. All jokes aside, I'm really grateful to my brother and my sister-in-law. They live in Lisbon. It's like midway between here and India, so I often go there. They cheer me up. My brother's completely the opposite of me. He's so chill and doesn't care about anything. And it's really useful to have someone like that in my life. And lastly, I want to thank my parents. They've done so much for me. I can't even begin to describe it at this point. And I'm just so grateful. And I know they're probably watching. My dad might be asleep, but I know my mom is probably watching me still. So I thank you and love you so much. And I'm happy to be there very soon back in India. And lastly, this is my dog. But stick around to celebrate, hopefully soon after that call.
And I think with that, the committee has to work. So